Hello viewers, today we explore a disturbing case of forensic science. Welcome to Decoding Death, the science of the postmortem interval. When investigators first step into a crime scene, they are usually greeted by total silence. But to the trained eye of a forensic expert, that victim is actually still speaking. One of the absolute most critical questions in any murder investigation is the timeline, specifically calculating the exact time of death. It is this fascinating, somewhat dark intersection where biology, chemistry, and pure investigative rigor all meet.
So imagine for a second that you were stepping right behind that yellow crime scene tape. A body has been found in a completely silent, undisturbed room. There are no witnesses around, no security cameras to check, and absolutely no murder weapon left behind. How exactly do forensic experts make a silent body speak? How do they read physiological and biochemical clues to reveal the exact timeline of a crime? Well, they do it by turning the human body itself into a biological clock, literally reading the subtle, inevitable changes that start happening the exact moment the heart stops beating.
In the world of forensic pathology, this biological clock we're talking about is known as the postmortem interval, or PMI for short. It is the scientific estimation of the time that has actually elapsed since a person's death. Now, it's super important to understand that PMI isn't like looking at an exact minute on a watch. It doesn't work like that. Instead, it is a highly analytical estimation range. By establishing this specific window of time, investigators can do things like validate a suspect's alibi, narrow down their list of potential killers, and build a rock-solid, legally binding timeline of events for the courtroom.
Section 1. The first 24 hours.
To really understand how this works, we've got to look at the immediate aftermath of death. This is that critical window where the earliest biological clocks start to tick. While we're alive and well, our bodies maintain a baseline internal temperature of roughly 98.6 degrees Fahrenheit. That's about 37 degrees Celsius. But the moment death occurs, this thermal regulation just completely fails. This kicks off a process called algor mortis, which really just means the cooling of the body. Gradually, the human body begins losing heat until it perfectly matches the ambient temperature of the crime scene. By carefully measuring this loss of heat, forensic experts can actually start winding back the clock.
Mathematically calculating this cooling rate requires a lot of precision. Medical examiners will typically use a highly sensitive thermometer to grab a core temperature reading, usually from the liver or rectally. Then they apply what's known as the Glaister equation. This basically estimates that the body loses roughly 1.5 degrees every single hour. And this equation is highly accurate, but really only within the first 18 to 24 hours. Investigators have to be incredibly meticulous here, though. Variables like the victim's body mass, the thickness of the clothing they were wearing, the air circulation in the room, or even if the body was immersed in water, all of those things can drastically speed up or slow down this cooling rate.
Now, as the body is cooling, another phenomenon kicks in, rigor mortis. After death, the human body stops producing ATP, which is the energy molecule required for your muscles to relax. Without ATP, the body's actin and myosin muscle fibers physically lock right in place. This stiffening usually starts up within 2 to 4 hours, hits full rigor mortis at about the 12-hour mark, and then it slowly disappears between 24 and 48 hours as the muscle tissues actually begin breaking down. Here's why that matters so much. If a detective finds a body with a disturbed rigor pattern, say an arm rigidly fixed in midair instead of resting naturally on the floor, it is absolute proof that the killer came back to the scene and moved the body after rigor had already set in.
The third pillar of this early postmortem interval is livor mortis, also known as lividity. Once the heart stops pumping blood, gravity just takes over. The blood begins to pool in the lowest points of the body, starting in as little as 30 minutes. By 2 hours, you'll see a noticeable purple discoloration appearing on the skin. And between 8 and 12 hours, this lividity becomes permanent, or what they call fixed. This brilliantly illustrates a key forensic strategy. If a body is found lying on its back, but the fixed purple lividity is on its chest, investigators immediately know the crime scene was staged, and that the body was repositioned hours after the actual murder. Even the specific color of this pooling can hint at foul play. Carbon monoxide poisoning, for instance, leaves a very distinct cherry red hue behind.
Section 2. The stages of decomposition.
But what exactly happens when a body isn't found immediately? As we move past that critical first day, those initial forensic indicators start to fade, and we enter the much darker long-term reality of human decay. The body undergoes 5 very predictable stages of decomposition, effectively acting as a long-term postmortem calendar. First is the fresh stage, spanning days 0 to 3, where decay is mostly just unseen internal bacterial activity. Second is the bloat stage, roughly days 2 through 6, where internal gases accumulate and cause swelling.
Third is active decay, days 5 to 11, which is marked by a really rapid tissue breakdown and a strong odor. This is followed by advanced decay, lasting up to 25 days, and finally, the dry remains stage, where only the skeletal structures are left behind.
So, the really crucial point here is that this timeline is definitely not set in stone. The environment is the ultimate wild card in criminology. Forensic experts have to account for external factors that dictate the exact speed of these 5 stages. Ambient temperature is for sure the most critical factor. Extreme heat drastically accelerates decomposition, while freezing temperatures can literally preserve a body for months. Investigators also have to carefully analyze humidity levels, whether the victim was buried beneath the soil or left out on the surface, whether they were in water, and of course the extent of any animal scavenging.
Section 3. Insects and chemical clues.
When environmental factors start to obscure the timeline, the investigation shifts focus away from the macroscopic changes in the body and turns toward nature's stopwatch, forensic entomology. Within literally minutes to hours of a death, insects, primarily blowflies, colonize the remains. They act as a living clock. These insects follow a highly predictable life cycle. They lay eggs, which hatch into larvae, then transition into a pupa stage, before finally emerging as adult. By measuring the length of the larvae, identifying the exact species, and cross-referencing all this with local temperature models, forensic entomologists can reliably estimate the post-mortem interval for bodies that have been decomposing for more than 2 days. We can also build our timeline through chemical pathology. When the external environment is just too chaotic, forensic experts will actually extract a sample of the vitreous humor, that's the clear fluid inside the eye. After death, potassium cells begin breaking down and leaking into this fluid at a very steady, measurable rate. Lower potassium levels indicate a shorter time since death, while higher levels indicate a longer time. And because this fluid is protected safely inside the eye, this chemical timeline is far less affected by environmental temperature shifts. This allows for incredibly accurate estimations spanning from 6 to over 100 hours post-mortem.
Section 4. Advanced Postmortem Science.
While these classical methods definitely form the foundation of criminology, modern investigations have evolved to look at death on a molecular level. Today, researchers track the degradation rate of DNA and RNA, which acts as a microscopic molecular clock. They also utilize microbiome analysis, tracking what's called postmortem microbial succession, basically the specific shifts in bacteria inside the body after death. And much of this groundbreaking science is backed by controlled observational studies conducted at specialized facilities right here in the U.S., most notably the University of Tennessee's Anthropological Research Facility, which is widely known as the Body Farm.
But what happens when a body has been missing for years? When all the soft tissue is entirely gone? Forensic anthropology is deployed. These specialized experts analyze skeletonized remains, carefully reading the weathering patterns on the bones and analyzing the chemical interaction between the skeleton and the surrounding soil. This incredible science allows investigators to estimate a time of death, even for victims who have been hidden away for decades.
Section 5. The multidisciplinary forensic approach.
In the real world of crime scene investigation, no single clue works in isolation. To build an undeniable case that will actually hold up in a court of law, investigators must bring everything together into an analytical climax. This is the multidisciplinary time-of-death estimation model. U.S. forensic experts know that relying on just one single factor is dangerous. Instead, they integrate classical pathology, looking at temperature and muscle changes in the first 48 hours, with chemical analysis of the vitreous humor up to 100 hours. If the victim has been undiscovered for days, entomology takes over. For weeks or years, they rely on decomposition and skeletal weathering. By layering these sciences over one another, the overlapping data points eliminate the outliers and build a precise, undeniable timeline of the crime.
Ultimately, time-of-death estimation is a multi-layered forensic process that brilliantly blends biology, chemistry, and environmental science. It's exactly how investigators reconstruct the truth, validate alibis, and ensure that those responsible are brought to justice. Science truly gives a voice to the dead, translating the silent evidence into the truth. Comment your thoughts on this video. Also, subscribe to us to see more videos like this in the future. See you in the next video.